A single blood test can tell you what is happening in your body today. But what if the real secret to aging is not the result itself? What if it is the direction that result is moving? A study published yesterday followed people across roughly 20 years and repeatedly measured thousands of proteins circulating in their blood. The researchers were not simply asking which proteins are higher in old people than young people. They asked something much more important. How does the same person's blood change as that person grows older? The study followed 1,298 people from middle and later adulthood into older age, measuring their blood at 4 different time points. Across the study, researchers profiled 10,776 protein markers. Additional data brought the total number of people analyzed across the research to 2,621. What they found challenges the way biological aging is usually measured. Some blood proteins remained remarkably stable for years. Others changed much more quickly. And most importantly, when a person's protein trajectory began drifting away from the pattern expected for healthy aging, that deviation predicted a higher risk of death. In other words, aging may not be captured best by one biological age number. It may be captured by watching whether your body is staying on its own healthy trajectory or beginning to move away from it. That distinction could eventually change how we detect disease before symptoms appear. To understand why, think about how medicine normally uses blood tests. Imagine your glucose level is measured today. The result falls inside the normal range. You go home thinking everything is fine. But suppose the same measurement has been slowly climbing for 10 years. 5 years ago, it was near the bottom of the healthy range. 3 years ago, it was in the middle. Today, it is approaching the upper limit. Technically, every individual measurement may still look normal. The trajectory tells a different story. Proteins may work the same way. The blood contains thousands of proteins produced by organs, immune cells, blood vessels, muscles, and other tissues. Some are enzymes. Some carry signals between organs. Some regulate inflammation. Some help maintain the extracellular matrix that supports tissues. Others appear when cells are injured or stressed. Because blood circulates throughout the body, it acts like a constantly moving information network. If the liver changes, some proteins may change. If the immune system becomes chronically activated, others may rise. If neurons, blood vessels, or muscles are under stress, some of that biology can also leave molecular traces in circulation. Researchers have already used this idea to build proteomic aging clocks. In one major 2024 study, scientists measured nearly 2,900 plasma proteins in more than 45,000 UK biobank participants. They identified 204 proteins that could predict chronological age with high accuracy. They then reduced the model to just 20 proteins while retaining about 95% of its age prediction performance. People whose protein profile looked biologically older than their calendar age were more likely to be frail, have poorer physical and cognitive function, develop multiple chronic diseases, and die earlier. That sounds impressive, but there was a major limitation. Most biological aging clocks are built from snapshots. One person is measured at 50, another at 60, another at 70. Scientists compare them and infer what aging looks like. But that is not the same as watching one person travel from 50 to 70. Imagine trying to understand how a city changes by photographing different cities at different ages. You might learn a lot, but you would learn something different by photographing the same city every 5 years for 2 decades. That is what makes the new Nature Health study unusual. Researchers repeatedly examined the same individuals over time. And the first important discovery was that proteins did not all behave the same way. Some had relatively stable individual patterns. If a person naturally had a relatively high level of one of these proteins, they might remain comparatively high years later. Other proteins were far more variable. That matters for biomarker development. Suppose a protein changes dramatically from month to month or year to year because of infection, medication, temporary inflammation, or some other short-term event. One measurement may provide a misleading picture of someone's long-term biology. A more stable protein may be a better marker of an individual's underlying physiological state. But stability itself is only part of the story. The researchers were also able to define expected longitudinal patterns. Instead of asking only, how old does this blood sample look? They could ask, is this person's blood changing in the way we would expect it to change? That is a very different question. Imagine two 65-year-olds. Both receive the same biological age estimate today. But one person's protein profile has been changing slowly and consistently for 15 years. The other's changed rapidly during the last 4 years. A snapshot might call them identical. A trajectory would not. And this may be where personalized aging starts becoming genuinely useful. The researchers found that deviations from expected protein trajectories were independently associated with mortality. This suggests that disruption of biological stability may appear before the final clinical outcome. The body may begin losing homeostasis. Homeostasis is the ability to keep internal systems within functional ranges despite constant disturbances. Your blood sugar rises after a meal and then comes back down. Body temperature changes and is corrected. Inflammation activates when needed and then resolves. Blood pressure adapts when you stand. Muscles repair themselves after stress. A young, resilient body does not remain perfectly unchanged. It changes and then returns toward balance. Aging reduces that resilience. The body may take longer to recover. Signals remain abnormal for longer. Different systems begin drifting away from their previous equilibrium. This is why biological aging might eventually be measured not only by where a marker sits, but by how well the body keeps that marker stable. The concept is similar to watching a tightrope walker. A skilled performer constantly moves. Small adjustments happen every second. What matters is not standing completely still. What matters is repeatedly returning to balance. As aging progresses, those corrections may become slower and less precise. Blood proteins could provide a way to observe that loss of balance. Some proteins identified in previous aging research help explain why this could matter. GDF15 is one example. It is a stress-related protein that tends to increase with age and is associated with numerous chronic diseases and mortality. Other proteins in established aging panels reflect inflammation, extracellular matrix remodeling, neuronal damage, hormone regulation, and energy balance. These signals do not represent one aging pathway. They represent many systems speaking at once. And recent research has made the picture even more complex. A 2026 Nature Medicine study analyzed more than 7,000 plasma proteins in over 60,000 people and estimated the biological age of more than 40 different cell types. The researchers found that aging was highly uneven. Around 20 to 25% of people showed accelerated aging in at least one cell type. A smaller group showed accelerated aging across many cell types simultaneously. And the identity of the aging cells mattered. Extreme aging of astrocytes in the brain was associated with substantially greater Alzheimer's disease risk in genetically vulnerable people. Extreme aging signatures in skeletal muscle cells were associated with later risk of ALS. Among smokers, accelerated aging of respiratory epithelial cells added additional lung cancer risk. The message is becoming difficult to ignore. There may be no single biological age. Your immune cells can age differently from your muscle. Your brain-supporting cells can age differently from your lungs. And even inside the same person, these trajectories can change over time. That means a future blood test may look less like a number saying, "You are biologically 58." Instead, it might look more like a dashboard. Your vascular aging trajectory is stable. Your immune trajectory has accelerated. Your metabolic signals started drifting 4 years ago. Your neurological markers remain younger than expected. That would be far more useful than one dramatic biological age score. Because a measurement becomes medically valuable only when it tells us what to do next. But we are not there yet. The new study does not create a clinical test you can order today to predict exactly how long you will live. The participants came from specific cohorts, and protein measurements depend on laboratory platforms. A blood protein can also change because of infection, medications, body composition, kidney function, cancer, or many other conditions. Association with mortality does not prove that the protein itself causes aging. A protein may simply be reporting damage somewhere else. That distinction is crucial. If smoke rises from a building, measuring the smoke can tell you something is wrong. Removing the smoke does not necessarily extinguish the fire. The same problem applies to aging biomarkers. Lowering an old protein does not automatically make a person younger. Before a protein becomes a treatment target, researchers need evidence that changing it actually improves health. There is another limitation. 20 years sounds extremely long for a biological study, and it is. But aging unfolds across an entire lifetime. The researchers mainly followed people from middle and later adulthood into older age. A person's molecular trajectory may have begun changing decades earlier. Diet, exercise, smoking, infections, pollution, sleep, medications, obesity, and chronic stress may already have shaped the proteome before the first sample was collected. So these blood trajectories are not a complete biography. They are a 20-year chapter. Still, the study represents an important shift. Most longevity research asks how old the body looks. This research begins asking how the body's aging process is moving. Speed matters. Direction matters. Stability matters. And sudden deviations may matter even more. This also changes how we should think about repeated health testing. One blood pressure reading can be useful. A year of blood pressure measurements reveals more. One weight measurement provides information. A 10-year weight trajectory tells a different story. One glucose result may be normal. A slowly rising glucose trajectory can reveal metabolic deterioration before diabetes is diagnosed. The same principle may eventually apply to molecular aging. The future may belong not to the most complicated one-time biological test, but to repeated measurements that reveal when a person's normal pattern begins to change. There is also an encouraging side to this. A trajectory is not the same thing as destiny. Some biological markers respond to physical activity, metabolic health, smoking cessation, weight change, medication, and disease treatment. Scientists still need intervention studies to determine which proteomic aging trajectories can actually be changed. But identifying the moment a system begins drifting could create something medicine rarely has. Time. Time to improve blood pressure before vascular damage becomes severe. Time to improve metabolic health before diabetes develops. Time to investigate unexplained inflammation. Time to preserve muscle before frailty appears. Time to treat disease earlier. That may ultimately be more important than knowing whether a laboratory algorithm calls you biologically young or old. Aging is not one photograph. It is a movie. For years, scientists have been trying to judge the entire story from a single frame. Now, after following thousands of blood proteins through 2 decades of human life, researchers are beginning to watch the film. And the most important signal may not be how old your blood looks today. It may be whether your biology is still following a stable path tomorrow.